Well, hello everyone, thanks for having me here. Today I'm going to talk about benchmarking single-cell RNA-seq preprocessing workflows. So basically, when we are trying to analyze a single-cell RNA-seq datasets, there are multiple steps involved. So firstly, we really start from the raw FASTQ files, and we will process into a cell gene count matrix, and this is what we define data preprocessing here. And afterwards, we really apply normalization methods to remove the influence of the technical bias. And sometimes we apply imputation method to deal with the sparsity of the counts. And often it depends on different aims. We will apply the collection method to find the cell state or cell type, and sometimes to find a slightly different stages of the development, we apply trajectory analysis. And also, if you have multiple samples, you apply the integration methods. And as single-cell RNA-seq becoming more and more popular, many methods have been developed at each step, and there are some available benchmarking study which would compare those methods, but they mainly focus on the steps on the downstream, which would be the parts after normalization, and leaving out the data preprocessing. So in this way, we think that a benchmarking study would be helpful and needed. So first, I will talk about the steps involved in preprocessing. So here we only focus on the UMI-based preprocessing workflows. UMI here, it's unique molecular identifiers. It's different from cell barcode. Cell barcode tells you the origin of the race of the cell of its cell, which cell it comes from, but the UMI is tagged to the cell before the PCR. In this way, you would count the transcript without influence on PCR amplification. And this strategy is what they used in this field. So what we get there will be two FASTQ files. The first one will contain the information of the cell barcode and UMIs, and another will contain the information of the cDNAs. And firstly, we usually just tag the cell barcodes to the head of the cDNA. And after that, based on different alignment tools used, we sometimes use traditional alignment like this, or some fancy new pseudo alignment tools like Listo or Silman. And afterwards, we need to deal with the UMI. As I mentioned, you would use it to remove the amplification bias. So you will collapse this UMIs, which is called duplication here. And while you're doing this, you might encounter some base error. And there are many existing strategies to deal with it. Some would use Hammond at a distance, or some choose to ignore it, and some even apply some sophisticated strategies. And afterwards, reads in separate cells would be counting to reads all the genes, and you will get a cell by gene count matrix. So we know that there are many steps involved in preprocessing, and to make it more convenient for the users, there are many built-up package preprocessing workflows. So here, for example, like the CellRanger, which is a very popular workflow developed by 10x Genomics, and like Cellman Eleving, which is a tool developed based on Cellman alignment free tool. And also like Optimus, which is developed by Human Cell Atlas. So using this package preprocessing workflows, you would just need to give the input, which would be the FASTQ files, and it would deliver count metrics for you. And there are also another workflow, which is assay pipe. It is available in Bioconductor, and it can process data from multiple platforms, such as 10x and Dropseq. And since it's implemented in R, so it will be easily to use together with other Bioconductor packages, such as Gator or scRNA, for the downstream analysis. So having this many package workflows, you must be wondering which one is better. Like it would deliver more accurate biological information, or which one run faster to save your time, and you're gonna make your decision. So before the comparison, there's another important aspect I want to mention is that for a benchmark study, you need to choose the good data sets. So in order to find which method is best, we need to know some kind of ground truth in the data. Here, let's three kinds of data sets. So the first is a real data. It would be the actual sequence, the real sample, which is what we usually see in a research paper. It will contain much biological information, but it would be extremely difficult to know the truly ground truth. Whereas for the simulated data sets, the underlying logic would always be the mathematical models, and we usually add the truth to them by ourselves, so we know the truth exactly well, but they will be less similar to the real data sets. And between them, there are some designed well-controlled data sets, which will contain cells like unknown cell line cells, and the designer would incorporate some experimental design, like to mix them with design proportions. And in this way, this kind of data set both contain good biological and information and ground truth. So they might be a good choice. So I borrowed a series of control data sets from this paper. It uses 5 lung cancer cell line cells, and some of them were processed by the CellSeq2 platform, which is a plate-based single-cell platform. And I select 2 design from it. The first one is an RNA mixture, which would contain a bunch of pseudo cells generated with pooled RNA mixed with listed proportions. And another would be simply mixing the single cells from different cell lines. So this dataset would provide us solid ground truth either from a plate annotation or the genetic information from them. And I also included a set processed by 10x version 2 and 10x version 3 with the same file, 5 cell line cells. And I also included the data which contain more biological complexity, which is a mouse lung tissue data set from the tabula muris. So here, first list the data sets I just showed you I would use. And next, these data sets are delivered to the pre-processing workflows. And these are the names of the workflow I choose to evaluate. I would both directly assess the quantification of the row count matrix, and I still want to try to explore the impact of the preprocessing on the downstream analysis with the help of another wonderful package, CellBench. So CellBench does a job which would automatically run the combination of the methods. For example, if I have 1 preprocessing workflow and 2 normalization methods and 2 cluster methods, it would generate 1 multiplied by 2 and then multiplied by 2, you would totally get 4 results. So in this way, you don't have to write a very complicated for loop, and this would be super convenient. So finally, we're going to the results. So first, for the 10x dataset, I tried to draw a density plot for the total counts per gene. And I find it's quite interesting. So for all the preprocessing workflow, they tend to show a bimodal trends here. And we could find that for cell ranger, the pink one, it shows more genes at the higher peak and less genes at the lower peak, which means more detected genes would have more counts. And well, for the other methods, they displayed a bit different. And for cholesterobusto, it displayed a third intermediate peak here. And I investigated if there were any characteristics of the genes at each peak. And I find that for the higher peak, there will be higher percentage of protein coding genes, which would be about 95%. While for the lower peak, only about 30% of them were protein-coding genes. So I guess this might caused by the sequence similarity between the pseudogenes and protein-coding genes, because for CellRanger, it recommended users to use a curated reference, which will only contain the link RNA and the protein-coding genes. So in this way, the pseudogenes are excluded. So from this plot, I was wondering if it might be sensible that the CellRanger recommended users to use the curated reference, because in this way, it would to some extent alleviate the impact caused by multi-mapping. But it is really sensible that, does it really make sense that remove the pseudogenes would give a better result? What if there are some signals contained in the pseudogenes, and does it matter? So I generated a BCV plot to find the answer. So here, this is a definition of the BCV, and in other words, it would tells you the proportion of gene expression are attributed to the variability, which means a higher value would mean more variability between a gene expression. In other words, there are more biological noise. And usually a BCV plot will start with a high BCV value, and as the abundance increase, the BCV would tend to be asymptotic. Here, we first look at the protein coding genes. All the cons developed by those preprocessing workflows showed expected similar trends, except for the three. They have more variability for the low expressed genes. Well, for the pseudogenes, despite of this one, the salmon alvin, the light green one here, it still show high BCV value even with abundance increase. And surprisingly for the SC pipe and the UMIs and the Optimus, they show the expected trends. And interestingly, for them, the pseudogenes displayed even less variability compared to the protein coding genes with a lowly expressed genes. So we might conclude that pseudogenes do capture some biological information. So I want to see what would it matter, would it influence the final clustering performance or not. So here, we apply normalization and clustering and get the combination of the performance, and then evaluate them with a tailored matrix. And here is our result show for ARI, which would measure the similarity between two data clusterings. And we fitted a linear model using ARI as a dependent variable, and also use experimental design and preprocessing workflows and normalization methods and clustering methods as corollaries. And so the positive coefficient would indicate that the method is positively associated with the ARI here. So in this way, higher coefficient value would mean better performance, and lower would mean worse performance. You could find that here, SC pipe, CellRanger, and dropsic pipe always influence positively. And also comparing preprocessing to other steps, we could find that preprocessing is quite influential. And we need to mention that previously we said about the CellRanger, it would use a curated reference. And here for the dropsic pipe, it shared most of their steps similar, except that the dropsic pipe would use the whole reference, not the curated one. So we might draw a conclusion that when you're using curated or the field annotation, one provide strongly different performance. And if you're trying to analyze the data and you are not that interested in pseudogenes, you might could use some modified reference, and in this way it will be easier for you to annotate the cells. And thanks for the lab members, and thanks for listening.